Hello students, today we will learn about microbial production of amino acids. So let's begin. The first commercial production of amino acid, glutamic acid, was started in Japan in as early as 1908. It was Ikeda, the Japanese scientist, who first identified that glutamic acid in the form of monosodium glutamate possess tasty enhancing properties. Taste enhancing properties. In the early days, monosodium glutamate was extracted from the vegetable proteins that may be from wheat and soya. It was only in 1957 the large scale industrial production of MSG by using microorganisms commenced. Today, commercial production of amino acids is one of the biggest industries world over with an annual increase in the demand by about 10%. Glutamic acid continues to be largest producer among the amino acids, followed by lysine, methionine, threonine, and aspartic acid. We will discuss all these terms, these acids. Okay, so let's begin from amino acid production. General consideration. What are the general consideration? So, some general considerations on commercial applications of amino acids, their production method, and development of strains of microorganisms for improved amino acid production we will now discuss. Now, let's begin. Let's start from commercial application of amino acids. Amino acids have a wide range of applications. The propionate use of amino acids is now we will discuss. Okay, first is food industry. In food industry it is used 65%, and in feed additives that is used 30%, and pharmaceuticals it is used 5%. The individual L amino acids along with their production method are, and uses are now we will discuss. Let's know about the food industry. Amino acids are used either alone or in combination as a flavor enhancer. Monosodium glutamate is the most frequently used in food industry. Glycine and alanine also enhance taste and flavor. Tryptophan in association with histidine acts as an antioxidant to preserve milk powder. For the preservation of fruit juices, cysteine serves as an antioxidant. Aspartame, a dipeptide, that is aspartyl phenylalanine methyl ester, produced by a combination of aspartic acid and phenylalanine, is about 200 times sweeter than sucrose. It is used as a low calorie artificial sweetener in soft drink industry. There are certain essential amino acids that are deficient or limiting in plant proteins. These include lysine, methionine, threonine, and tryptophan. Addition of the deficient amino acids improves the nutritional quality of human food as well as animal feed. Thus, breed enriched with lysine soy products supplements with methionine are of better nutritional value. Methionine added soybean meal is a better feed for pigs and other animals. Now let's know about the pharmaceutical industry. Amino acids can be used as medicines. Essential amino acids are useful as ingredients of infusion fluids for administration of patients in post-operative treatment. Now let's know about chemical industry. Amino acids serve as starting materials for producing several compounds. Glycine is used as a precursor for the synthesis of glyphosate phosphate or a herbicide. It is a herbicide, while 309 is the starting material for the production of azithrionum. That is another herbicide. Polymethyl glutamate is utilized for manufacturing synthetic leather. Some amino acids in the form of N-acyl derivatives are used for the preparation of cosmetics. Now let's know about the methods for production of amino acids. The industrial production of amino acids is carried out by one or more of the more process. There are three processes. Let's know about these three processes. First is extraction, chemical synthesis, and microbial production. Let's know from extraction. What do we mean by extraction? Amino acids are the building blocks in protein structure. The proteins can be subjected to hydrolysis and the requisite amino acids can be isolated. Example: cysteine, tyrosine, leucine. Now, let's know about chemical synthesis. Chemical synthesis results in a mixture of D and L amino acids. Most of the amino acids required for commercial use. Now, let's know about the microbiological production. For the large scale production of amino acids, microbiological methods are employed. There are three different approaches. Let's know about the approaches. First is direct fermentation method. Amino acids can be produced by microorganisms by utilizing several carbon sources. Example: glucose, fructose, alkanes, ethanol, glycerol, propionate. Certain industrial byproducts like molasses and starch hydrolyzed can also be used. Methanol, being a cheap carbon source, is tried for amino acid production but with limited success. Now let's know about conversion of metabolic intermediates into amino acids. In this approach, the microorganisms are used to carry out production. Example: conversion of glycine to serine. Third is direct use of microbial enzymes or immobilized cells. Sometimes resting cells, immobilized cells, crude cell extracts, or enzyme membrane reactors can be used for the production of amino acids. Amino acid hydrogenases from certain bacteria like Bacillus megaterium can be used for the amination of alpha keto acids to produce L amino acids. Example: alanine from pyruvate, leucine that is from alpha keto isocarboic acid, and phenylalanine from phenyl pyruvate. Immobilized cells or enzyme membrane reactors can be used. Enzymes or immobilized cells are also employed for the production of several or other amino acids: tryptophan, tyrosine, lysine, valine. Strain development for the amino acid production. Let's know about the strain development for amino acid production. The metabolic pathways for the synthesis of amino acids by microorganisms are tightly controlled and they operate in an economical way. Therefore, a natural production of amino acids is a rare occurrence. Some strains that excrete certain amino acids have been isolated: glutamic acid, alanine, valine. In order to achieve an overproduction of any amino acid by a microorganism, methods have to be devised for the elimination of the metabolic regulatory or control processes. In fact, several amino acid producing microorganisms have been developed by mutagenesis and screening programs. There are some major ways of strain development. Let's know the process. The ways. In fact, several methods are combined to successfully develop a new strain for producing amino acids. First is auxotrophic mutation. These mutations are characterized by a lack of the formation of regulatory end product, that is repressor or regulatory effect. The intermediates of the metabolic pathways accumulate and get extracted. Second is genetic recombination. Mutants can be developed by genetic recombination for overproduction of amino acids. Protoplast fusion in certain bacteria is used for development of hybrids. Example: Cornybacterium, glutamic, glutamicum, and Bacillus flavum. Now let's know about recombinant DNA technology. The classical techniques of genetic engineering can be used for strain development. Strains with increasing activity of rare limiting enzymes have been developed. In one of the technique, E. coli and cloning vector PBR322 were used to increase the genes for the production of amino acids. Example: glutamic acid, lysine, phenylalanine, valine. Now let's know about the functional genomics, that is a new approach. An analysis of genomes from the wild and mutant strains of microorganisms will help in creating improved strains. Once the entire sequence of the chromosomes in the organisms, example C. glutamicum or E. coli, is established, efforts can be made to carry out genetic manipulations for efficient overproduction of desired amino acids. Chip technology can be used to detect new mutations and consequently the fermentation process. Now let's know about L-glutamic acid. L-glutamic acid was the first amino acid to be produced by microorganisms. The original bacterium, Cornybacterium glutamicum, that was first used for large-scale manufacture for glutamic acid continues to be successfully used even today. The other important organisms employed for glutamic acid production belong to genera Microbacterium, Brevibacterium, and Arthrobacter. All these organisms have certain morphological and physiological characters comparable to C. glutamicum. Biochemically, glutamic acid producing bacteria have a high activity of glutamate hydrogenase and a low activity of alpha ketoglutarate dehydrogenase. They also require the vitamin biotin. Now let's know about the improved production strain. Several improvements have been made, particularly in C. glutamicum, for improving the strain to produce the excrete and more of glutamic acid. These include the strains that can tolerate high concentrations of biotin and lysozyme sensitive mutants with high yield. Now let's know about the biosynthesis of L-glutamic acid. The pathway for the synthesis of glutamic acid with glucose as the carbon source we will now discuss. Glucose is broken down to phosphoenol pyruvate and then to pyruvate. Pyruvate is converted to acetyl coenzyme A and phosphoenol pyruvate by the enzyme phosphoenol pyruvate carboxylase can be independently converted to oxaloacetate. Both these carboxylation reactions are quite critical and require biotin as the cofactor. The next series of reactions that follow are the familiar citric acid cycle, that is, we can say Krebs cycle reactions wherein the key metabolite, namely alpha ketoglutarate, is produced. In the routine citric acid cycle, alpha ketoglutarate is acted upon by the enzyme alpha ketoglutarate dehydrogenase to form succinyl coenzyme A. For the production of glutamic acid, alpha ketoglutarate is converted to L-glutamic acid by the enzyme glutamate dehydrogenase. This enzyme is a multi-mer each subunit with a molecular weight of 49,000. The reducing equivalents in the form of NADPH plus H plus are required by GDH. They are generated in the preceding reaction of Krebs cycle while converting citrate to alpha ketoglutarate. The supply and utilization of NADPH plus H plus occurs in a cycle fashion. through the participation of two enzymes, namely isocitrate dehydrogenase and glutamate dehydrogenase. Theoretically, one molecule of glutamic acid can be formed from one molecule of glucose. In practice, the conversion efficiency of glucose to glutamic acid was found to be around 70%. Now let's know about the regulation of glutamic acid biosynthesis. The essential requirement for glutamic acid production is the high capacity of the supply of the citric acid cycle metabolites. This is made possible by an efficient conversion of phosphoenol pyruvate as well as pyruvate to oxaloacetate. Thus, there are two enzymes, phosphoenol pyruvate carboxylase and pyruvate carboxylase, to efficiently produce oxaloacetate, while there is only one enzyme, that is pyruvate dehydrogenase, for the formation of acetyl coenzyme A. Certain microorganisms which have either phosphoenol pyruvate carboxylase, example E. coli, or pyruvate carboxylase, that is example beta B subtilis, are not capable of producing glutamic acid to any significant extent. Glutamicum has both the enzymes and therefore can replenish citric acid cycle intermediates through oxaloacetate while the synthesis of glutamic acid occurs. Another key enzyme that can facilitate optimum production of glutamic acid is alpha ketoglutarate dehydrogenase of citric acid cycle. Its activity has to be substantially low for good synthesis of glutamic acid, as is the case in C-glutamic acid. Further exposing the cells to antibiotics, that is penicillin and surfactants, reduces the activity of alpha ketoglutarate dehydrogenase, while glutamate dehydrogenase activity remains unaltered. By this way, oxidation of alpha ketoglutarate via citric acid cycle can be minimized, while the formation of glutamic acid is made maximum possible. Let's know about the release of glutamic acid. Glutamic acid is synthesized intracellularly, and therefore its release or export is equally important. It's now appears that there is a carrier mediated energy dependent active process involved for the export of glutamic acid. There are several ways of increasing the membrane permeability for exporting glutamic acid, that is biotin for limitation, addition of saturated fatty acids, addition of penicillin, use of oleic acid oxotrophs, use of glycerol or oxotrophs, addition of local anesthetics, addition of surfactants between 40, that is 2 in 40. The effect of biotin deficiency is facilitating the release of intracellular glutamic acid has been worked out. Biotin is an essential cofactor for the biosynthesis of fatty acids. Due to a limited supply of or deficiency of biotin, fatty acid biosynthesis and consequently phospholipid synthesis is drastically reduced. As a result, membrane formation is a defective which alters permeability for an increased export of intracellular glutamic acid. It is found that there is an alteration in the membrane composition of phospholipids in oleic acid and glycerol oxotroph mutants. This facilitates release of intracellular glutamic acid. The knowledge on the membrane permeability of glutamic acid is successfully exploited for increased industrial production of glutamic acid. Now let's know about the production of glutamic acid, requirements and influencing factors. The industrial production of glutamic acid is influenced by carbon sources, nitrogen sources, growth factors, pH and oxygen supply. The now relevant aspects we will now discuss. Production of glutamic acid requirements and influencing factors. In this, we will discuss. First is carbon sources. Either refined, that means glucose, sucrose, fructose and maltose, or undefined, that means sugar beet, molasses, sugar cane molasses, etc. Carbon sources are used. In countries like Japan, acetate, inexpensive, is utilized. Other substrates like alkanes, ethanol, and methanol are less frequently used. Now let's know about nitrogen sources. Nitrogen sources. The concentration of ammonia is very crucial. The concentration of ammonia is very crucial for converting carbon sources to glutamic acid. However, high concentration of ammonia inhibits the growth of organisms. The beginning of fermentation, ammonium salts and a low concentration of ammonia are added. During the course of fermentation, ammonia in aqueous solution is continuously fed. In this way, pH can be controlled. Besides continuous supply of nitrogen sources, sometimes urea is also used as a nitrogen source since glutamic acid producing bacteria processes urease that can split urea and release ammonia. Now let's know about growth factors. Biotin is an important growth factor and its concentration in the medium is influenced by the carbon source. For instance, is For instance, a supply 5 microgram of biotin per liter medium is recommended if the carbon source is 10% glucose, while for acetate as a carbon source, the biotin requirement is much lower, that is 0.1 to 1.0 microgram per liter. Addition of L-cysteine in the medium is recommended for certain strains. Now let's know about supply of oxygen. Oxygen supply should be adequately and continuously maintained. It is observed that a high oxygen concentration inhibits growth of the organisms, while a low oxygen supply leads to the production of lactic acid and succinic acid. In both instances, glutamic acid formation is low. Now let's know about process of production and recovery. Some important information on the production of glutamic acid by Brevibacterium di- divaricatum. Now we'll discuss. First is carbon sources. Here carbon sources is glucose, that is 12%. Nitrogen sources is ammonium acetate, that is 0.5%. pH is 7.8. Temperature it is 38 degrees centigrade. Period for fermentation, that is 30 to 35 hours. Yield of glutamic acid, that is 100 gram per liter medium. And as the fermentation is complete, the cells are separated. The culture growth is passed through anion exchangers. The glutamic acid bound to the resins is eluted in sodium oxide, sodium hydroxide, NaOH, while the ammonia released can be reused. With NaOH, glutamic acid forms monosodium glutamate, which can be purified by passing through anion exchanger. MSC can be subjected to evaporation and crystallization. Now let's know about L-lysine. Lysine is present at a low concentration in most of the plant proteins. Being an essential amino acid, supplementation of plant foods with lysine increases their nutritional quality. L-lysine is predominantly produced by Corynebacterium glutamicum and to some extent by Brevibacterium flavum or beta lactofermentum. Now let's know about biosynthesis of lysine, L-lysine. This metabolic pathway is also involved in the formation of three other amino acids, namely methionine, threonine and isoleucine. As the glucose gets oxidized by glycolysis, phosphoenol pyruvate and pyruvate are formed. Both these metabolites can be converted to oxaloacetate a key component of citric acid cycle. On transamination, oxaloacetate forms aspartate. The enzyme aspartate kinase converts aspartate to aspartyl phosphate, which latter forms aspartate semialdehyde. Aspartate semialdehyde has two fates: the biosynthesis of lysine and formation of three other amino acids, that is methionine, threonine, and isoleucine. When hemiserine hydrogenase acts on aspartate semialdehyde, it is diverted to the synthesis of three amino acids. The enzyme dihydrodipicolinate synthase converts aspartate semialdehyde to piperidine 2,6 dicarboxylate. There are two distinct enzymes: succinylase variant catalyzes four-step reaction and dehydrogenase variant catalyzes a single-step reaction that can convert piperidine 2,6 dicarboxylate to DL diamino pimelate, which latter forms L-lysine. Now let's know about the regulation of L-lysine biosynthesis. There is a process for the production of lysine. Let's know about this process. First enzyme is used, aspartate kinase. This enzyme is controlled by feedback inhibition of the end product. Three isoenzymes of aspartate kinase have been identified, one replaced by L-methionine, the second one replaced by L-threonine, and thus L-threonine and L-isoleucine, and the third one being inhibited and replaced by L-lysine. The amino acid sequence and replaced by the amino acid sequence and structure of aspartate kinase have been elucidated, and by genetic manipulations it has been possible to create mutants that are insensitive to feedback regulation by L-lysine. Dihydrodipicocolinate synthase. Let's know about this enzyme. This enzyme completes with homoserine dehydrogenase to act on aspartate semialdehyde. Overexpression of dihydrodipicolinate synthase has been shown to increase the production of L-lysine. Now let's know about the succinylase and dehydrogenase variants. The conversion of piperidine 2,6 dicarboxylate to DL diaminopimelate is carried out by these two enzymes. At the start of the fermentation, dehydrogenase variant predominantly acts and later succinylase variant comes into picture for the biosynthesis of L-lysine. Now let's know about the role of DL-diaminopimelate. This amino acid and intermediate precursor for the synthesis of L-lysine is also required for the synthesis of a tripeptide, which is part of the peptidoglycan of cell wall. The activities of both the enzymes succinylase and dehydrogenase that form diaminopimelate, in short DAP, are important for the production of L-lysine and for the proper formation of cell wall structure. Now let's know about improved production strains. Based on the biosynthesis synthetic pathway and the regulatory steps, certain improvements have been made in the strain of glutamicum and B flavum for overproduction of lysine. Mutant organisms resistant to lysine antimetabolites, example B aminoethyl L-lysine, a mutant strain with an altered enzyme aspartokinase so that it is not regulated by end product inhibition, a strain with a decreased homoserine dehydrogenase activity so that diversion for the synthesis of methionine, threonine, and isoleucine is minimized. A strain with reduced citrate synthase activity to lower the occurrence of the citric acid cycle. Now let's know about release of L-lysine. The export or release of L-lysine from the cells into the surrounding medium occurs through a lysine export carrier protein. It is a transmembrane protein with six segments that participate in lysine transport. The exporter system is very efficient, active process to export large quantities of intracellular lysine. Let's know about the production process of L-lysine. The most commonly used carbon sources for lysine manufacture is molasses, starch hydrolysate or sucrose. The other sources like acetate, ethanol or alkenes are used to a lesser extent. The nitrogen sources are ammonium salts, gaseous ammonia, protein hydrolysates are added to supply certain amino acids, L-methionine, L-homoserine, L-threonine. The protein hydrolysates also supply growth factors such as biotin. A time course graphic representation for the formation of lysine we will now discuss. As is evident, a continuous supply of glucose or other sugar is required for sustained production of lysine. Under optimal fermentation conditions, the yield of lysine in the form of L-lysine HCl is 40 to 50 gram per 100 gram carbon sources. There are different recovery processes for lysine depending on its application. An alkaline solution containing about 50% L-lysine can be obtained after biomass separation, evaporation and filtration. A crystalline preparation with 98 to 99% L-lysine can be obtained by subjecting the culture broth or to ion exchange chromatography, evaporation and crystallization. Both these grades of lysine are suitable for supplementation of feeds. Now let's know about the L-threonine. L-threonine is manufactured industrially by employing either E. coli or C. glutamicum. With the mutant strains of E. coli, the product yield is better. Now let's know about biosynthesis of L-threonine. Some of the reactions of this pathway are common for the biosynthesis of L-lysine and methionine. Besides isoleucine, starting with aspartic acid in a sequence of 5 steps, threonine is produced. Now let's know about regulation. The regulatory reactions in E. coli for L-threonine biosynthesis have been elucidated. Three isoenzymes of aspartate kinase separately inhibited by the end products have been identified, one by L-threonine, one by L-methionine and one by L-lysine. For the two isoenzymes of homoserine dehydrogenase, one inhibited by L-threonine and other by L-methionine are also known. A gene threonine ABC that encodes three polypeptides. One polypeptide possesses the activity of kinase and homoserine dehydrogenase. The second homoserine kinase and third threonine synthetase in E. coli has been identified. Now let's know about improved production strains. The efficiency of the producer strains can be increased by creating E. coli mutants with high level expression of the gene THR-ABC. Further, mutants with minimal production of L-isolysine also result in high yield of L-threonine. Now let's know about production process of L-threonine. The culture medium containing glucose or sucrose, yeast extract and ammonium salts is adequate for L-threonine production. The sugar feeding with the sugar feeding has to be continued for good yield. About 60% of the carbon sources. The downstream processing for the isolation of L-threonine consists of coagulation of the cell mass, fertilization, concentration of by evaporation and crystallization. Now let's know about L-phenylalanine. Both E. coli and C. glutamicum can be used for the production of L-phenylalanine. The biosynthetic pathway is quite complex, and an interesting feature is that the same pathway is responsible for the synthesis of all the three aromatic amino acids tyrosine and tryptophan, and besides phenylalanine. The synthetic pathway commences with the condensation of erythrose L-phosphate with phosphoenol pyruvate to form deoxyarabino heptulosonate phosphate, in short DAHP. DAHP in the next series of reactions is converted to corismate, which can form L-tryptophan. Corismate mutase converts corismate to prephenate, which forms L-phenylalanine through the participation of prephenate dehydrogenase. Prephenate also serves as a precursor for the synthesis of tyrosine, the genes responsible for the formation of the regulatory enzymes of L-phenylalanine have been identified. By employing genetic manipulations, strains for improved production of L-phenylalanine have been developed. Now let's know about L-tryptophan. There are different ways of synthesizing L-tryptophan: chemical, enzymatic, and fermentation methods. At present, large-scale manufacture of tryptophan is carried out by using the enzyme tryptophan synthase of E. coli. Tryptophan synthase combines indole with L-serine to form tryptophan. Indole is available for petrochemical industries, while L-serine can be recovered from molasses during sugar refinement. Mutant strains of E. coli with high activity of tryptophan synthase have been developed for large-scale manufacture of tryptophan. Now let's know about direct fermentation process. Tryptophan can also be produced by fermentation employing C-glutamicum or E. coli. Mutant strains of both these organisms have been developed for increased yield of tryptophan. Now let's know about mutant strains for overproduction L-tryptophan. The production of tryptophan by C-glutamicum was increased by introducing a second gene encoding anthranilate synthase, a key enzyme in its biosynthesis. Further gene encoding other important enzymes like deoxyribinoheptulosonate phosphate synthase and anthralinate phosphoribosyltransferase were also be modified. The result is that the pathway becomes insensitive to feedback inhibition by end products, leading to an overproduction of L-tryptophan. Now let's know about L-aspartic acid. There is a growing demand for aspartate as it is a component of aspartame, besides its use as a food additive and in pharmaceutical preparation. The preferred method for aspartate production is enzymatic in nature. The enzyme aspartase converts fumarate and ammonia to aspartate. Although this reaction is reversible, aspartate formation is favored. Aspartase. The aspartase of E. coli is used. It is a tetramer with a molecular weight 1,96,000. This enzyme is quite unstable. Immobilization of aspartase is polyacrylamide or carrageenan that enhances the stability of enzyme is commonly used. Immobilized E. coli cells with good activity of aspartase are also used for aspartate production. Finally completed. Any questions regarding this, you can ask me in the comment box. I will definitely answer the questions, and please kindly subscribe my channel to know more updates. Thank you.